Okay. So anyway, it's a great honor to have Ben Boda here. I'm going to move the slides for him when he asks me to. Let me move him back. He came at a special time. He could tell you more, but he was an undergraduate and a graduate student of the biological sciences department. And I started in 2003, so I've got to know Ben for quite a long time. And Ben can tell you more about his story, but he's a cancer survivor like myself. And I think that helped kind of develop a special bond between us because I was going through cancer when I met him, and so I think we developed a little bit of a bond through that, as well as our love for nature. So we definitely did quite a bit of fishing together and adventures. And so we developed a kind of a real family-like atmosphere in our labs at times, and Ben was definitely a big part of that. And one other thing I want to mention, so Ben, like I said, he got his start with caterpillar saliva, like many in my lab, and then they go on and do some amazing things, whether they stay in plant-animal interactions or go on into medical field or teaching field. Ben is a great example of that success, and he also was involved with the corn to ethanol research experience or internship when he was with Western that we were able to do with I guess it's called the Illinois Rural Institute with Dr. Chris Merritt, back when there was some money and there was some interest in that. Obviously, ethanol and fermentation still an important field, but that was a big plus to get him started as well. Ben's from the St. Louis area, and like I said, it's always been great. I've been catching up with him from time to time. I think he's often tries to provide internships for our students or jobs too. So, Ben, I think I don't want to steal your thunder too much, but I'm really excited to have you here. I don't know if you can, if you like to unmute your image.
Yeah, that's fine. Yep,
He's a serious fisherman. Outfished me every time we went out. There should be a video button. You see where you can set up your video camera? There's just a little.
That's you.
Oh, cool. All right, so I'm going to step down and let you take over. And thank you so much for everything. You've been an amazing student over the years, and I appreciate it.
Okay, can you all hear me?
Yes.
Yes.
Okay.
Yes.
Okay. Good. I'll start my journey. This is my journey through the scientific field, from being at Western, starting at Western, and now to where I am. I work in Pfizer research and development, primarily, not manufacturing, which we are kind of thrown into a little bit right now. Not quite a year now with research and development, and also the production of the COVID vaccine. I guess next slide. Okay, I started Western Illinois in 2001, the fall of 2001, and I was a biology student. My primary focus was animal biology. I don't know if Western still has the biology where you could go into botany or something like that, but basically, for the most part, my classes focused on, animal perspective, a lot of field work, taxonomy, as far as identification or classification of things, and whether you went into a certain thing like ichthyology or something like that, or and things like that, you could learn about a certain organism, how they work, do this and that, but primarily focused on animals. I didn't really have too much of a background in plants, but photosynthesis, just from the general biology classes when you first started at Western, the 100 level or so classes and everything. So the one thing I did find helpful, even through, maybe you didn't necessarily do that, do or work with it, or some of the labs, not only in biology, but I also had a chemistry minor. So I also was in chemistry. I took inorganic chemistry, organic chemistry, forensic chemistry. Main thing I was interested in was forensic chemistry, was DNA. Some of the PCR we would do, this, that. I mean, it's gotten a lot, if you go out in the field, it's got a lot more advanced in the last, 10, 15, 20 years. So it's a little bit different from what they do, but some of the old school techniques in PCR with gels and things such as that. Next slide. Okay, I actually graduated in 2006, and I started grad school in the fall of 2006, and I eventually joined Dr. Musser's lab. Primarily, didn't have really too much of a plant biology, so I actually background. So I actually took some classes in basically plant physiology and things such as that, just to learn more about this and that. And also, by far, labs helped in those classes, and things, different studies we did in those classes helped out. Primarily in Dr. Musser's class, we focused on plant and animal interactions, and basically a lot of our experiments had to do with caterpillars. Of course, I don't deal with caterpillar spit anymore, but I used to a lot, whether that be extracting caterpillars, saliva glands, and wounding the plants and painting them on the plants, or mechanically wounding the plants and comparing them to a plant that maybe hadn't been damaged in any way whatsoever, or however you wanted to do that. My master's basically focused on the analysis of variants from a plant that had been fed on by whiteflies, or actually tomato plants had been fed on by whiteflies, versus plants that were non-wounded and everything like that. So just to see if there was any difference in the genes, and then you had to identify genes and figure out what they were. And then you have the different things with plant and animal defenses and stuff like that, which I think one of Dr. Musser's big ones years ago was glucose oxidase and everything. We did a lot of plant growth, whether it be out in the field or in the small chambers. I guess they're still there in the basement of where you could set the lighting and everything down in the basement of the biology department. And we used to grow, tobacco, corn. I think we had soybeans a couple times, probably tomatoes. And I know we did several lab experiments out in the field with caterpillars, putting them on plants and trying to harvest them, which was always fun to try to figure out where they migrated to. Usually they would stay in pretty much the same spot if they had food, but it was always a challenge. It did a lot of RNA purification, not only extraction, but purification. For the most part, run it on a spectrophotometer at nucleic acid. So you're running it at a wavelength of 260 nanometers, since it's a nucleic acid, not a protein. Then we hybridize these chips, or go through the process of creating complementary DNA and putting them on cDNA microarray chips, and then with two different dyes, and basically seeing which where the different genes were, whether they were stimulated or suppressed by the chip areas turning, examining the chips, little spots on the chip that would turn red, green, or you get a yellow or some kind of color like that. You could analyze those chips, and then you could figure out if this gene was suppressed or stimulated, or nothing happened, or compared to a control group. A lot of times we would have compare a non-wounded plant to a mechanically wounded plant, or a non-wounded plant to a plant that maybe a caterpillar fed on. So Uh, when I was at Western, they had a program. I don't know if it still exists with National Corn to Ethanol Research Center, which is on the campus of SIUE. They used to, when I first started working for them, they said they didn't have a lot of affiliation with the university, but I think they really do. Matter of fact, the new logo says at SIUE, which was very convenient for me because I'm actually from Edwardsville. So it was like going back home and working back home. Matter of fact, there's a picture of the building there. That was actually closer a drive for me than whenever I used to drive to high school. So it was very convenient. Corn to ethanol, they had a small scale pilot plant there to do trials and stuff, though they usually were only running if they had a company come in and say, hey, we're going to pay you, we want to do this run, or we want to try this or try that. But most of the time, the only time we were running was for test runs and things like that. And you'd be running like what you would find in a typical ethanol plant. Versus we did research, we would get a lot of samples. Illinois Corn Growers Association would a lot of times supply samples every month of whatever their dry distillers grains and solubles or their ethanol and this and that. Any kind of sample you could think of, the corn they got, you could measure different things with. We would run detections, various compounds on HPLCs, do Carl Fisher to measure ethanol content, a lot of standard wet chemistry, whether that be your pH, moisture, read ash. One of the things I was involved in was measuring glucose and starch, pretty much by defatting the corn, the ground up corn, and doing that, and then doing an analysis and figuring out how much of a composition that was, to give them kind of an idea of, oh, this is how much starch was in and whether it be a certain corn strain they were growing or what, I don't really know. But we would do things in the labs too, measure density. Dextrose equivalent was a big one to measure the amount of reduced sugars present in a certain sample of once they had everything ground up and in a fermenter and growing. So it was a very, outside of academia, for those of my first kind of real experience, they did run like a company, but at the same time they were research, and they were all on grants for the most part, supplied by grants and that. But it's a very, very good start to learn how to do certain things, and especially certain methods and how they did things and this and that, versus going from a research more in an academia place versus a kind of private company somewhat. Next slide. Okay, my actual first job outside of there, I left there. They were having, Corn Ethanol was having a lot of financial problems, grants and stuff were falling apart, and I ended up leaving there and have a friend who was at Sigma Aldrich in downtown St. Louis. I worked at their Cherokee site, which is actually right by where Anheuser-Busch is. If anybody knows where that is, it's not actually too far. Matter of fact, in this picture, whenever I flew it down, you could actually see Anheuser-Busch in the background. But Sigma, I worked in a GMP facility, basically, which is fun because it was my first experience with that. I don't particularly like it. Everything has to be documented. Everything, you can't really make changes on the fly. Like whereas where I'm at now at Pfizer, I can. We primarily manufacture of APIs, which are active pharmaceutical ingredients, and basically we were a contract manufacturer. We'd have certain companies that come in and saying, we want you to do this, or I were actually, after I was there for a little while, I took over for 3 projects, and basically they were all produced because of for a Abbott Vascular who and I found out they had a big presentation, and because I knew who they were for, but what they actually did, and they actually went into breathing apparatuses for premature infants that helped expand the lung capacity. So probably in neonatal hospitals or areas. So a lot of what I did at Sigma was based on a lot of it was chemical reactions. We were a non-animal, so anything animal that was produced with animals could not be used. All of our stuff had to be, of course, everything we used had to be sampled, sent off to quality control, and then released by quality assurance and everything like that. One of the things I was involved with there, I was in charge of our microbial monitoring program, which at times could be a nightmare. We do plate analysis, particle analysis through certain machines and stuff like that, and measuring all that. And one of the things I got to work with was non-conformances, which I hope I never have to work with again in the field. But definitely a learning experience in how to do things, and work in a GMP environment is definitely something, you know, that helps you out along the way. I was a chemist there at Sigma, and one of the things I would do is involved in all the approvals and everything with anything as far as operation procedures, changing things to batch records, and then going through, I think I had a list of about 7 different people who had to sign my approvals. And a lot of times you would get ones that didn't quite understand the field, or especially in the QA department, which quality control or quality assurance had never worked in the field. So that was always fun. As far as chemical reactions.
Dr. Musser, you're going to have to mute him.
Yeah.
I'll figure out which one it is. Thank you, Lindsay.
Okay. Yeah, a lot of that involved a lot of chemical reactions and things like that, and a lot of it was based on purification, whether it be filtering stuff through. We had a process that required charcoal filters that were 6 foot tall, and we had 4 of them, and we'd go through one to the other to all lined up. Or a lot of the stuff was based on distillations to purify, or you could do recrystallizations. We had one process that had 3 different recrystallizations and stuff like that. But yeah, definitely a learning experience doing that, and working not only working with other people within your own company, but with other actual pharmaceutical companies. So it was very much a test, to say the least. Last slide. Okay, I currently work, I started working in late 2012 for Pfizer, research and development. Basically, we're referred to as BRD, or Biological Research and Development, and we're based in Chesterfield, Missouri, which is about 20 miles west of St. Louis, or downtown St. Louis. Primarily we are a non-GMP pilot plant, which I like because I make changes if I see something. I work in both upstream and downstream operations. We do both microbial and mammalian cells, and we basically upscale things from labs for the most part. Labs have very small things they work with, and they see potential. They send it to us, and we put it on our schedule and try to do everything. And most things, I would say, we're definitely the backbone of research and development of any pharmaceutical company, but I would say probably about 95% of the things I work on would never go to the market, or you would never even hear about. So it's rewarding, but very challenging. Both mammalian and microbial are fairly similar to work with, and really the only difference is the lysis of the cells, because with mammalian cells, they're very touchy. You have to watch out what you do with them. Even pumping them through a pump can damage the cells. When we harvest them, we use centrifuges, and a lot of times that will actually lyse the cell, versus your microbial cells. You're either going to have to chemically lyse or go through a homogenizer for the most part to break the cells open. One of my big things that I do here is, and I've been trained, I've actually went out to a little bit west of Boston for Nova Biomedical, which is we run our NovaFlex, which every time we sample things with mammalian side, we could run a cell density, seeing how many cells are in it, any kind of gases, nutrient metabolites such as glucose, lactate, and a bunch of other things. So I primarily take care of that, which can be challenging. As I said, I went out to Boston a couple of years ago and trained with them for about a week of actually where Nova's at. But so that is primarily most of our things we deal with here, and it has been lately within the last, I would say, 6 to 7 years, is a big thing, is working on MABs, or also known as monoclonal antibodies. Basically, there's a lot of, you'll see them on commercials. One of the big ones, that you see all in the commercials all the time, I think Abib makes it, is adalimumab. And obviously the last 4 letters in the word is MAB, is monoclonal antibody. So just for, if you ever see a commercial, that's what that stands for at the very end of things. It's been becoming more challenging. The market has definitely, especially with research and development, because it used to be just a single MAB. Now they're coming up with bispecifics, trispecifics, where you're getting growing up, harvesting numerous MABs and putting them together, conjugating them together with, usually a lot of times it's with a DMSO or some kind of linker, some kind of enzyme, something to link the bonds and make whatever. That's way above my level. Some of the senior scientists, principal scientists, and stuff like that get to deal with all that. And it's something else. And then as of about a year ago, everything came down. We were working like we normally did, and we've become somewhat, with all the construction and everything that I'm doing over the last year or so, got involved with the making the plasmid DNA for the COVID vaccine, and that's actually made here at the Chesterfield site in the pilot plant. And there's a couple other departments involved in doing so. But basically, it serves as a spike protein on your basically your SARS-CoV-2 or CoV-2 virus, which is the virus that causes COVID-19. Basically, that makes it recognizable to the immune system. It's because it's known as the novel coronavirus, meaning that the immune system does not detect it or have a way to fight it, because it doesn't know how. And basically, the mRNA is basically sending instructions to the cells to basically, this is what it is. So just like a flu shot or something like that, recognition more than anything. That's produced right here. Eventually, there's 3 different sites which are currently involved with that. Here in Chesterfield for the plasmids, they send it to Andover, Massachusetts, which is about 25 miles north of Boston. We have another site there, and they basically transcribe that into mRNA, and then that gets eventually shipped to Kalamazoo, Michigan, which is kind of in the southwestern part of Michigan. And basically they encapsulate the mRNA and lipids and package it into vials. And I think that's where They have a freezer farm there, and I know it's in that. I know there's pictures of it that in the article. Dr. Musser attached along with this, and yeah, they basically package everything. They have, I think, like thousands and thousands of feet. It's a huge warehouse of just freezers, and they package it there and put it in the freezers and eventually ship it out to wherever it goes. So I had the final slide. I was just going to talk. I know people looking for success in the field, and just a couple things of advice for people. And this is just for me working in the industry for when I have, and different people, dealing with different people, and just in general, I've had talks with my bosses about certain things too, and or certain ones have recommended things to me. And some things of advice I advise a lot of people to do is don't burn the bridges behind you per se. You never know. That it could be unpredictable at times, depending on where you're at. Now, I'm not worried. I have pretty good job security here, but there's other places that will turn around and after a couple of years and decide, hey, we're gonna clean house or sell the site or something like this, and either you relocate or you kind of lose your job or get packaged out or whatever. So it's good, you have, whether that be even people that worked for the company at one time or whatever, or a past job you had within the field. I actually know somebody who I worked with in the past at both Sigma and Pfizer, who's not the greatest at doing that, and he is actually has applied to different departments, and a lot of times they won't even bother to look at his resume. And I know he went somewhere and got let go because of his behavior and everything else like that, and really doesn't have anywhere to come back to if he would like to. Another one, we kind of mentioned before, but connections with people. You'll probably work with somebody, no doubt, that's not going to stay at the same place, whether that be a chemical company, manufacturing site, anywhere. But so it's always good to keep connections. Matter of fact, I can hear somebody making a bunch of noise right next to me, and it's one of my former Sigma colleagues, and he works here now, and he works in our CLD group, or cell line development, or we also call it cell banking group. And so it's always good to know people along the way, make connections, and let's say one day that you're looking for a job and somebody contacts somebody, or somebody contacts you and saying, hey, I think you'd fit in here really good. I talked to this, maybe I'm at maybe they're at management level, or maybe they know somebody within the company that can get you a job somewhere. The last two things are actually two things one of my former bosses when I first started at Pfizer told me. Basically, I'd have monthly meetings with him in his office and things like that. And one of the big things he said to me was, you're out there, he said, gain all the experience you can here. Because he said, because that's, experience is something we can't take away from you. Somebody can't take that. Hey, I worked with this, or I did this, or even in the interview, going somewhere and saying, hey, I did this, I did that. I've worked with this, that. So that's always a positive. And the other thing he told me one time is, in an interview, a lot of us get a little bit tense and everything with everything, and try to do the best you can in an interview. And he basically told me, he said, with your experience and everything, make them want you, make them hire you. Don't make them go back and have a discussion. Well, do we want this person or not? Or hey, is there there any other people that we just interviewed that we may want instead? I mean, when I actually applied for Pfizer, I had enough experience working with in the field with all my experiences, not taken from WIU all the way on to where I was the last one who basically did an interview. It was a 3-hour interview. I interviewed with in front of 6 different people. I got a tour of the place, and then I got pulled in the office for basically just talk with the boss who I was applying for the job at, and he basically told me, he goes, I want you here now, this, that. I was the last one to interview. He basically hired me on the spot. So that's always a good thing. And that is about it. Any questions?
Yeah, I got a couple for you. Oh, okay. So you were talking about how experience is a really good thing. I mean, how can it translate over even to a field that doesn't necessarily relate? Like, I'm working on my master's right now and doing research in cancer, specifically ovarian, but I'm looking more towards the animal medical field. So I'm wondering how that would translate, for example.
Any experience. I mean, even as little as any laboratory experience, anything like that, anything just learning along the way. I mean, if I would have came probably straight out of college, there's no way I'd have the job I have right now. So a lot of it's a learning experience over time.
Okay. Did you have direct work with the COVID vaccine yourself?
What did you say? What was that again?
Did you have direct research on the COVID vaccine itself?
Does my research apply to anything with the COVID vaccine? Currently, what I do, no, nothing. It's all, matter of fact, most of the people here, we kind of got this. This actually, we work alongside with bio and tech, and basically this has been classified as Project Lightspeed because most pharmaceutical, to get approval and anything like that, I've worked with people who've been here 30, 35 years or more, and they said the fastest we've ever seen one from start to getting FDA approval has been about 5 to 6 years, which this one took about 8 months.
I think Nick's
So,
oh, sorry. I just wanted to make sure. Did you get, Nick, did you have a question?
Yeah. How was your current work different from work that you've done before, or how has your career changed over the course of it?
how is it? Primarily, I'm not in manufacturing. I'm in research and development, and at some point, a lot of it's the same. It's just now we are GMP, and we have to follow certain protocols, follow this, follow that, versus making changes on the fly was usually a pretty typical thing. It's not untypical of similar things we've done in the past by far, but it's still a lot of change as far as the process and everything else like that. So
One of the things I thought was really cool was that it shows, one of the things Ben really said was really need to keep your people skills. It's not enough just to be a lab rat. You really need to be able to interact with people. That kind of take home messages, and then keep your skill level high.
yeah, definitely. It's good people skills, good to make connections, whether it be anywhere in the field and everything like that. Matter of fact, the guy I can hear him through the wall next to me, and he's he actually trained me at Sigma. Of course, I told him, of course, you train me what to not to do. So kind of jab at him at times, but yeah, he trained me at Sigma and everything else like that. And he actually came to Pfizer about 6 months before I did, and then it kind of helped me out, even though he worked in the actual clinical department, which actually went away. Most of those people were either went to Andover, Massachusetts, where the clinical moved to, or they were packaged out. So on retention and severance packages with the company. So another one stayed and migrated into different areas. This one actually migrated over to cell line development. There's a lot of, even within BRD, there's a lot of different areas. There's cell line development, there's analytical research and development, there's process purification development, and there's upstreams, and there's several other development groups within the BRD biologics. So
So, you had mentioned in your college career, you had started out as animal biology and then plant biology. How were you able to transfer that when you moved towards the chemistry and what you do now? Did you find that the knowledge was easily transferable, or can you speak to that a little bit more?
It was definitely something different going from animal, not really having a full idea with plants, going back a lot of different classes, studying this, that, knowing, finding stuff out, looking things up and everything. But as far as my jobs with afterwards, corn to ethanol was definitely a different thing, using some of the instruments such as HPLCs and things such as that you might use in the lab. We have them here. We have the labs up above this building, and we have a pilot plant off to the side, and we have different groups within that. We have a laboratory support group. That help set things up for some of the scientists upstairs. We have people that we have a solutions group or a buffer group that makes certain solutions because we need things to run through, whether it be ultrafiltration devices, chromatography skids, media to grow things with. So yeah, so little things along the way usually help, but a lot of it's slowly a learning curve. We're getting to a new area, such as, I worked with when I worked at Sigma, I didn't do anything that was growing cells or anything like that. So it was kind of new to me, and I slowly picked up on things. So kind of a learning curve, to say, with a lot of it.
How hard and long do you work things? I figure, it's just amazing what industrial science does. We got the fact that we got millions and millions of vaccines out there now. Is it just nonstop working and 12 hours?
Yeah, very much. Yeah, we just started our mammalian side up of our plant a couple weeks ago, which it's been that's going to be fun with trying to run with this COVID plasmid, and then on top of doing that. So I told them they're going to have to back off on some of it, or we're not going to be whoever's scheduling things. So it's it gets very interesting around here, and since and that's primarily just on research and development like we normally do. So producing it for whatever, looking at we send samples off to the labs and everything else like that. Or we do some of our work is team supply, which will grow something up, whether it be as small as something in shake flask or a wave bioreactor, would may only be like anything from a liter to maybe about 25 liters, and basically hand it off to people upstairs in the lab growing stuff or doing whatever, or basically growing things up and even taking them to a downstream process, and at some point getting to a certain point and handing it off to the lab so they can work more with it and they have more material, versus maybe growing something up and having a liter of it to have 25 liters of it. So
Ben, can you give kind of a general, what is kind of like an entry-level scientist make, and what does a senior-level scientist make, and how do they get vacation time, and how stressful does it get? I know this is unusual.
For the most part, you're usually scheduled. You usually have a certain schedule you stick to. I'm primarily in charge of second shift in the pilot plant, so I have two others right now that I oversee their work, and I also help them out I'm on the floor because they usually need my help because with things. So but usually there is a associate scientist, and that's usually probably right out of college. I definitely say the pay here is much better than it was at Sigma. And basically bump up. Usually if you're coming in here with a PhD, you're starting as either a scientist, possibly senior scientist if you get lucky. But it's it's different levels. There's different levels of jobs throughout the company, whether that would be not different levels as far as whether you're on a technician base or a T level versus an R level versus a management level. There's all different ones. So definitely, I enjoy working here. It's definitely rewarding. It can be frustrating at times, especially with R&D, knowing that probably the project you're working on isn't going to make it probably very far. And if it does make it to clinical stage, then you have the steps of the clinical stage, which to get to even FDA approvals. Which, with technology and everything, we're definitely moving in the right path to where it's getting better because we're catching things a lot sooner than we normally would. So, and just the ability to figure things out. Hey, we know this isn't going to work in the lab, so let's just scrap this, or let's try to do something different. We've had ones in BRD with they've gotten to certain points and they've just said, hey, great work with everything, but we can no longer go forward with this process. So
I had a question about the COVID vaccine, kind of. You were saying that it took 8 months for the whole process to go through for the COVID vaccine, whereas it took like 5 to 6 years previously. And my family has also read research about all of that, and they're kind of nervous to get the vaccine because of it. So how would I go about explaining to them that that's not something to be scared of?
It should just be sending instructions to the cells. I mean, it's a mRNA vaccine, so it should just be doing that. But I know they had it come through here, and I know they've said side effects could vary. Usually I've heard more people tend to have more side effects, kind of achiness, such as that, with the second vaccine versus the first. And we had some around here that, I just feel a little bit fatigued. I don't feel the greatest after the second vaccine, but all of them within a day or so, they're fine. So it's a new process, and
Okay. So do they take interns at Pfizer, or it's just experienced workers they take as workers there? Do they take interns? Do they train people?
yeah. Most of us coming into these jobs have had some kind of background, but a lot of it, especially R&D, a lot of it's new. And I mean, we have people upstairs. We have principal scientists, stuff like that. I work with one. I think he actually has a medical doctor degree on top of a PhD, and his specialty is in downstream operations and a lot of protein folding and things such as that. But a lot of it's just a learning experience over time, and working with certain things, and a lot of the scientists, some of in certain levels are involved in just they have a certain project or a certain part of a project, nothing else. So they get a lot of experience working with just a single part of a project. So versus I get the upstream, downstream, and everything else with it. So it can be challenging at times.
Yeah, Ben, thank you
Yep.
so much. Thank you so much for what you do. I would like to ask, what are the things that you find most challenging during your time at Sigma?
What did I find most challenging about my time at Sigma? One of my biggest problems with Sigma was the turnover. They didn't pay particularly well. By the time, it was very frustrating because by the time you get somebody new in, you train them, and by the time they weren't, even after a couple months after starting to train them, they were gone because they found a different job that would pay more. And I think since Merck took over, bought Sigma out, I think in 2014, everything's gotten better there. But that was probably my biggest frustration with Sigma is keeping people there, because a lot of times most of us moved on, and there weren't a whole lot of people that knew what they were doing. And that was the biggest problem I heard whenever I left. A little bit after, they would have people train online with everything with GMP for about 2 weeks. By the time you had to read everything, you got on the floor, and basically they said, somebody told me they were handing them operation procedures and saying go and do it. And I heard they had a lot of failures because of that. So that was my biggest thing with Sigma, with the problem and frustration and anything else. But so hopefully that all turned around when Merck started working, or Merck took over. So
yeah, thank you.
You said you worked on Pfizer for the upscale production for industrial use. How is making things for industrial use different from like laboratory use?
I do small things such as shake flasks and stuff like that, but definitely something a learning curve, I would say, because whenever I obviously was in the labs at Western, undergraduate and graduate, I moved to when I actually started working with National Corn Ethanol was my first experience with using. I would go into the pilot plant, and there was probably 6 different ports in which I would get samples off of or take samples from. So that was my first experience working with anything like that. And then I eventually, when I started at Sigma, was a small scale pilot plant, which I wasn't that familiar with at all. And basically a big learning curve also. And I think that's a lot with, usually you're going to jump around and try to gain experience, this and that. I mean, unless you're going to try to do like a PhD where a lot of, I've noticed with PhDs, we get them here, a lot of, I get notifications, join a WebEx meeting, hear this person's speech and everything like that for interviews. And that's normally what I tend to find is a lot of specialty, but yeah, basically for me, it's been a lot of a learning curve as you slowly. My background is mainly in downstream operations and not so much upstream. And I actually have somebody here who is an upstream expert who can help me out if I need it. But I can do some things in that just over time, learning and everything else like that. So.
Could you talk a little bit about the difference between upstream and downstream?
Yeah, upstream, you're gonna grow things in a reactor or something. We primarily use subs, which stands for single-use bioreactor. Kind of has a bag in it where you can hook up your probes. We have dissolved oxygen probes, pH probes, and the bags usually have pressure sensors in them and other things. And have a media grow our cells in it and everything like that, and supply the gases and everything, lines to it and everything like that. And then basically it's usually harvested with a centrifuge, and then from there it's a lot of times filtered, taken downstream. Downstream operations include anything from chromatography to ultrafiltration normally. We've had other things with certain projects, we have different things that come up, but and different things we do. So depending on what the labs maybe want to see with certain things, or if it makes it to the next level, what they can expect. But yeah, that's basically the difference. Basically, you're growing the cells to harvesting the cells, and then basically the downstream purification of the cells and everything. So.
So it seems pretty quiet. Does anybody else want to ask a question?
I guess I have a kind of a weird question about the vaccine itself, in the sense that I've been wondering biochemically, are the first dose and second dose any different than the other?
I don't know that because I don't do anything like that. My guess is it probably not. But yeah, I don't work with any. I just do the production of the plasmid, not any of the. I would say it's probably just recognition, overall amino recognition.
Can you tell them about the plasmid for some who might not know about?
Okay. What about the plasmid?
Well, this will be a quick synopsis of what the plasmid is in regards to.
Basically, you have circular pieces of DNA within the cells, basically that are basically plasmid DNA. They attach. What we do is we get it basically to attach to the SARS-CoV-2 virus, which SARS is a single acute respiratory syndrome, a virus, and basically that's what causes COVID-19. So basically, grow up, harvest the plasmids, linearize the plasmids, and then we ship them off, and then they go to Andover, transcribed into mRNA, which at Andover, Massachusetts, and then that's sent to Kalamazoo, Michigan, where it's basically lipid encapsulated and packaged into vials and put in freezers and eventually shipped out.
That's fascinating. So you can see how many hands are just.
Oh yeah, and we not. It's not only us. We have our cell line development and all ARD. Everybody, we send samples, test samples and stuff like that. Bioburden endotoxin samples on almost every step are sent out, and everything like that.
I think the romantic view of science is always that one scientist in the corner somewhere figuring everything out, but it's really army.
Yeah, definitely. Yeah, there are so many groups involved in this. So yeah, it's so many sites, different sites and people involved in this right now.
I'm just imagining all the coordination. And so to me, it's still amazing that there's something out, even though always people think it should be, might have different ideas about what the timelines be. To me, I'm always amazed that things work and people get things happening. So
yeah. Well, I got to go here in a second. I have to actually inoculate a fermenter here in about an hour. I got to get set up everything. There should somebody, I don't know what media outlet, but I heard Monday, and I heard rumors New York Times. I don't know if that has any weight to it or not, but I will, if I hear something or get anything from my boss, that article I sent you, the president of Biologics and Research and Development, who is here, sent that out. So, and he sends articles all the time out to us that he sees, finds, and whatever. So,
thank you, Ben, so much for coming and sharing your experiences and a little bit of your history. And it was really fascinating. It seemed like a lot of good questions came out of it. Thank you so much.
Yeah, I'll keep you updated if there's any jobs here, or they usually do internships, but I did not see that last year because of the COVID. So, but I'll keep you updated.
I don't know if we can do a little round of applause. I'm sure people are muted, but thank you so much.
Yep, thank you.
Take care.
Thank you.
Take care.